video. You have the link. You can open in YouTube. Um, and it is very nice summary what we have learned. I went quite slowly with the first part because it's important for you to understand. And if we cannot finish our lecture, it's okay. Yeah. So let's start with the video. Okay. So this is a summary of number one of the DNA structure. So we talk a lot about this. Is DNA is double-stranded helix and is bind together through hydrogen bond. You have learned about the Chargaff rules, about the number of the A and T is the same and C and G is the same. The double helix, DNA double helix is right-handed that you learn, and the directions is important. Go from 5' to the 3'. The two polymer of the double-strand helix is anti-parallel, and they are complementary, right? And so you, with this, we can move to the next part. Because so you learn about the 3D structure of DNA. But if you look at the nucleus, yeah, what you see under microscopy is look like this, or under electron microscopy is look like this. This is the nucleus, the dark part of the nucleus. Or this is the nuclear membrane. You can see that the close to the membrane, the structure is also something different. And you can see that the nucleus, they form somehow also compartment within the nucleus. Although there are no membrane to separate the space. What you can imagine is that we have an apartment or a house with several rooms, like the nucleus is the biggest room in the house, and within the room, you also have compartment, but you don't need walls, right? You can use something to make to separate the space without membrane, and it's the case with the nucleus. And you know that DNA are found in nucleus. It's highly compact here, and it's very well organized. But you cannot imagine how the double helix DNA is organized here within the nucleus. Do you know? So the length of the DNA molecules of the cells. Do you know how long is this? It's 2 meters. It's 2 meters. How can it organize in such a nucleus? The a nucleus is just about 6 to 10 in diameter. So it's much, much smaller, right? So the DNA, which is found in the nucleus, they have to be organized somehow. And this is why we know about chromosome. So the human DNA is packed into how many chromosomes? 46. There are 44 autosomes and 2 sex chromosomes, the X and Y. Yeah. And how can it be? How can the double-strand helix, what we learned, can be packed into such a structure like chromosome? Do you know which protein play here a role? Does somebody know?
Histone.
Yeah, right. So let's learn about histone. Before we go to histone, it's very important to mention that not all the nucleotides what we see in our DNA encode protein. The human genome just have about 30,000 genes. A bacteria have only 500 genes. Okay? Most of the human genes code for protein. And however, you can see that many segments. For example, here's the chromosome 22. Yeah, you can see this red spot and like the white spot. And when we have a closer look, you can see this part, they encode, this part is encode proteins. That means that RNA will be or mRNA will be transcribed on this part to make protein. This part they call exon. Exon is a part of the chromosome or the DNA which encode protein. However, there are many parts in between. They don't encode protein. This is called intron here. I think here you see better. It's exon and it's intron. These exon they encode, they contain the genetic information to make protein. And the intron here they don't make protein, but they also contain genetic information. There's there and what is the functions of the intron? If they don't encode for protein, they contain information for regulatory DNA. That means, for example, the promoter genes. Later, we can learn closer about the promoter genes. Because, as I said, that not all genes express at the same time. Can you imagine that all the cells in our body, we have the same DNA content, right? But we have so many cell types, right? So how can the cells know that what I should do, which protein I express? It means that the protein synthesis depends on cell type and also depend on the environment and signals that the cells get. So how can it be correlated? It's due to the regulatory DNA sequence. For example, the promoter to regulate the transcriptions of DNA. Okay, so it's important for you to know there's exon and intron in our DNA. The exon is the region which encodes information for protein synthesis. Intron contain many regulatory elements. Okay. So in total, there are about 3 billion nucleotides in our in the human genome. It's very large, and in 2004, people would be got the complete sequence of the human genome. It was very challenging to do the sequencing at this time. It took many years to translate the information from that they got. Nowadays, it's much more simple with the next generation sequencing technique, and also the algorithm that they use now is much better than before. So let's learn about the structure of the chromosome. The chromosome, as you saw previously, they look somehow different, right? They have different bands, they have different lengths, but what they have all have the same. They have a centromere here in the mid. You have centromere here, and they have telomere here in the both sides. So the structure at the two sides, at the two ends of the chromosome is called telomere, and the centrosome is here. What are their functions? So the centromere is the structure where the mitotic spindle attached to during mitosis. So the sister chromatid can be separated into the two poles of the divided cells. Okay? And the telomere is in the end of each chromosome. It contains of many repeated nucleotide sequences. So you can imagine that the telomere protect the chromosome from damage, because because you see that it's the positions where it's the most easiest to be damaged, right? To be get uh, uh, to get let's say to get some it's often that this the part get is break off or fused with other chromosome in cancer cells. So it's the most unstable structure. So you can imagine that during replications, because DNA replicate from 5' to 3', so from in this one end, the polymerase bind to the DNA and prolong the new synthesizing DNA strand. After each cell cycle, the telomere is shortened, getting shorter and shorter, because every cell in our body, through the life, they have divided for many times, and each time the telomere is shorter. It's just a few, let's say, it's few hundred maybe nucleotides gets shorter each cell divisions, but in total, so you can imagine that when we age, our telomere of the cells is shorter compared when we were young, and our chromosome also getting less stable. Okay, so the functions of the telomere is to protect the chromosome. Okay, and to ensure that the genetic information is contained in the middle of the so here within the in chromosome can be delivered from the mother cells to the daughter cells. So another structure that are found in all the chromosomes are the DNA replications origin. What is the DNA replications origin? Is where the DNA replication start. Each chromosome contains many DNA replications origin, because you know that we have 3 trillion DNA, 3 trillion nucleotide, right? So of this nucleotide or the DNA, the whole DNA of the cells has to be double when the cells divide, right? So in order, in order that it's take not far, if each chromosome just have one DNA replication origin, it will take forever, right? So in order to make this process faster, the cells have organized that there are many DNA replications origins that fire the DNA replications at the same time within this nucleus. Okay. So now let's learn about how DNA organized in a chromosome. For example, the chromosome 22 we learn about. So chromosome 22 has 48 million nucleotide pairs. And in the form of double helix, it will be 1.5 centimeter in length. But during mitosis, when the DNA condenses, it's just 2 microlit length. So of course, people will ask how DNA can be so condensed in a chromosome. And you guys have suggested, remember me, with a histone protein. Right. So another question is that the DNA can be very condensed, for example, during mitosis. But somehow, if you would have a look at the cells or nucleus during interphase between the two mitosis, you can see that the DNA are less dense, right? So even though this DNA is less dense, they also have to unwind in certain time point. For example, when the DNA replications origin is fire, right? In this time, the DNA polymerase have to be able to bind to the DNA replication origin to start the DNA synthesis. Or another example, when a gene should be transcribed into RNA to make protein. So the promoter regions of these genes have to be open for transcription complex, right? Okay. So the DNA packaging system has to be flexible enough to get, let's say, on-demand access to the DNA. When the cells need protein complex have to be get access to DNA in a exact reason where it's needed, right? The last question is that as I mentioned, that all the cells in our body, we have the same DNA structure. We have the same hereditary information. How can each cell know what is its functions and what they should do, right? It's not that easy. If the DNA is the same, what is the difference that we learn in these sections? So, if people unwind the chromosome structure, you can see this very, very typical chromatin structure. Chromatin. Do you know what this chromatin means? Chroma is color. Chroma is mean in Latin color. Chromatin, a substance which have color. Okay. So if you unpack the chromosome, so you can see this type of this typical structure of chromatin, and is 13 nanometer in diameter. So remember it. The chromatin structure, which is typically has 13 nanometer in diameter. Okay. If people stay up, so further unpack this chromatin structure, so you can see a structure like this. It's like a string with some like a small ball here. It's like pearl, you know. It's a seven, and so it's each one pearl connect with the on small bits. Okay, connect with the next one with a very thin fiber. Okay, this structure has a diameter of 10. So after that, when people separate DNA and protein here, they discover it. This car, this small bits here, is made of histone. And they know that histone and non-histone protein both interact with DNA to make their chromatin structure. So what about histone? Thus, this car, these bits as you see in under electron microscope. Oh, this is an histone car. This consists of 8 histone molecules, 2 histone 2A, 2 histone 2B, 2 histone 3, and 2 histone 4. Okay? The histone 2A and 2B interact with each other to make a Tetramer is that a molecule consists of 4 units. Okay? The 2 histone 3 and 2 histone 4 make up another tetramer, sorry. And these 2 tetramers, they make an octamer. This is the histone core. And the DNA, 147 nucleotide pair nucleotide, they wipe outside the histone core and to make this 10 or 10 or 11 nanometer in diameter. Okay? If you have a closer look, you can see that how the DNA interact with the histone core through its minor groove. Do you remember in the video they explained that protein can interact with the DNA in their major and minor group? And the major group is for a specific binding and the minor group is for non-specific binding or non-specific interactions. And as you see here, this histone is a structural protein. So in these organizations, that they expose the DNA expose their major group to outside, so other protein complex can easily interact with the major group of the DNA. It's quite interesting, right? You can also see the structure of the histone. The histone 2A, histone 2B, histone 3, and histone 4. They are closely related. They have more or less similar structure. All of them contain 3 alpha helix. The alpha helix makes a structure like a set, and 2 helix, 2 histone molecules interact together to make something like an X. And this is a dimer. And 2 of the histone 2A and 2 of histone 2B made together octamer, and 2 histone 3 and 2 histone 4 make another tetramer. And they make here an octamer, a histone octamer. As you can see that the cells are the end of each histone molecule, they stay show to the external environment so that they can exchange information or interact with other protein complex and to regulate the DNA functions. Okay? The histone compositions of the DNA is quite, let's say, it's not really flexible, but it's not, it can be changed. And this enzyme can change the histone, which are bind to DNA in on positions with a histone isotype, or variant. The histone variant may have different functions compared to the conventional histone. And it's also, with this, it changes the characteristic of the DNA in this region. Okay? So we have learned that the DNA wide about to the histone core to make the 11 nanometer structure. How can it organize from this 11 structure to the chromatin structure? It's nanometer, it's not micrometer. I'm sorry, yeah, for this mistake. So how can it make from this 11 nanometer to a 13 nanometer chromatin structure? It functions through histone 1. The histone 1 is not found in the histone core, but is connect the histone together. So the histone 1 is bigger than the other histone and is interact with both the other histone and DNA. It's like a clump, and it can tuck the histone core stick to each other to make the chromatin structure. Okay? This structure can still be condensed during mitosis using other protein. So let's have okay. Let me find the link for this and then we can have a quick look with this video and then we make a break again. Okay. Do you get the link?
Yes.
Yeah.
Okay. So let's start it. Yeah. So, just short a quick review what we have learned. So we have learned that DNA is packed into nucleosomes to create the 13 nanometer chromatin structure, right? Histone protein and also other non-histone protein play important role in packaging process and also modifications of the structure. Uh, in the histone core, you can find the histone 2A, histone 2B, histone 3, histone 4, right? And the histone 1 links nucleosomes to nucleosomes. So, what we have learned, how chromatin structure organized in the nucleus, right? You can see here in this figure that how can somebody describe the structure of the chromatin within the nucleus for me? Remember that chromatins mean color, and people, so scientists previously they look at the nucleus of the cells and see color structure there. So what did they see? I'm sure they see the nucleolus, right? Where the
Teacher,
Mhm.
I can see your screen.
You cannot see my screen. Oh, I'm sorry. Hmm. Sorry. Can you see it now? See this.
Not yet.
Mhm.
I haven't seen the slide yet.
Okay, 1 second. Let's try again. So let's show the screen again. Oh, oh. Okay. Since when it was lost?
I see the
It's
slide
lost
now.
for yeah. Slide nó biến mất từ bao giờ hả các em?
Em
Mất
nghĩ
từ
là
cái lúc mà
chắc
cô
từ
gửi cái link
kiểu video
video.
ấy ạ.
Ok, rồi thế thì cũng không quan trọng. Ok. So, could somebody describe how you see the chromatin structure in the within a nucleus? Please. Could somebody please describe it? What you see here? I'm sure that you see the nucleolus. It's a big dark structure here. It's the largest compartment within the nucleus. And what is the function of nucleolus? Do you remember?
Is to store the DNA and chromosome.
Mm-hmm. And the most well-known functions of nucleolus to synthesize what? A cell's deep compartment. The empty mean mitochondria.
DER and Timidoculus.
Mitochondria. Yeah, it's the place where mitochondria is synthesized. Okay. Sorry, sorry. The ribosome is synthesized. How can I make this mistake? The ribosome is synthesized. Okay. So, you can see the structure of the nucleolus is quite dark. Yeah, it's darker than the other part in the nucleus. So it is very obvious for people when they check the under electron microscopy, they can see the nucleus intimately. And do you notice that other part they also have very dark structure here? It's and you see where it also have quite dark structure. So color here is close to the nuclear membrane, right? And so people notice that the chromatin they localized in the nucleus in different compartments. They are heterochromatin and euchromatin. Heterochromatin they contain so heterochromatids they contain mostly non-coding regions. Most of the genes are silenced there. And when the cells they become active, they should be transcribed, they move to the euchromatin regions, where it's lighter under microscopy here. Yeah? So let's have a break until 11:05, okay? And after that, we can continue with the lecture, okay?
Okay.